Hey everyone, I'm Alex, and today we're going to walk through DNA replication. DNA replicates so that each new cell that gets produced during cell division gets an identical copy of the original genetic information from the original cell. Okay? Without replication, the daughter cells that are produced at the end of cell division wouldn't have the instructions that they need to function properly. I want to point out that the detailed understanding of DNA replication has led to some amazing life-saving medical treatments that can stop DNA replication in harmful cells such as pathogenic bacteria or human cancer. So as you can imagine, if we have an original cell that has harmful DNA, we don't want to go through the process of cell division and DNA replication to create more of them and then lead to tumor buildup and cancer. So it's worth learning about. Okay, let's get started. I think before I list the steps for you, it would be great to have an understanding of the key enzymes because otherwise you'll read the steps and you'll see these words and you won't know what they mean and it'll seem really overwhelming. Okay, so let's start here. Helicase unzips. When we have the two DNA strands, as you know, they're intertwined together. Okay, helicase unzips them by breaking through the hydrogen bonds that holds the DNA bases together. DNA primase is the initializer. DNA polymerase, which is the third one, is what actually builds the new DNA strands, but it can't do so without a primer. So primase makes the primer so that DNA polymerase can figure out where to start working. And it's worth noting that the primer that primase creates is made out of RNA. This is important later. Another thing that's important later is the need for glue in this process. Ligase is an enzyme that helps glue DNA fragments together. Okay, so let's do the first two steps first. Let's talk about how helicase unwinds the helix and separates the strands, and how primase creates the RNA primer so that RNA polymerase knows where to start building. DNA replication starts at a certain part called the origin, and usually this part is identified by DNA sequences. At the origin, helicase, the unzipping enzyme, which I've shown here in blue, comes in and unwinds the DNA. Now, when we unwind it and are left with these two strands, unwinded strands here, we don't want them to coil back up. So something called SSB proteins, molecules called SSB proteins, come and bind to the DNA strands to keep them separated. Topoisomerase is a molecule that keeps the DNA from supercoiling, and supercoiling needs to be controlled during DNA replication, and it can involve an overwinding of the DNA if topoisomerase didn't exist and supercoiling occurred. You really need the DNA strands to be separated for the next steps, which are shown here. So light green is primase, the enzyme that we spoke about, and dark green is the RNA primer that it creates. Okay. Primase will come in and make RNA primers on both strands. And as we said earlier, because without this step, DNA polymerase wouldn't know where to start. Before we move on to the next ones, these ones are a little bit more complicated. So before we move on to them, I want to refresh our understanding of DNA structure because having a good foundational understanding of DNA structure will make steps 3, 4, and 5 flow so much easier into your brain and it'll be a lot more seamless and your understanding will be much more thorough. So let's do that. Remember how we said DNA has two strands? So what we showed here, strand 1 and strand 2, okay? Strand 1 and strand 2. As you may know, they complement each other and they're anti-parallel. They complement each other because the nucleotides here, the base, sorry, these squares are not nucleated. This is a nucleotide, but as you can see in the key, we've got a base, which is adenine, guanine, cytosine, and thymine. This is the sugar, and this is the phosphate, okay? So they're anti-parallel because they go from a 5' to 3' direction and a 3' to 5' direction in orientation, but they complement each other because the base is here. The chemistry between them makes them attract each other, okay? So if this was an adenine, this would be a thymine because they're attracted to each other. Likewise, if this was a guanine, this would be a cytosine because those two attract to each other. Now, what do I exactly mean by 5 to 3 prime direction and 3 to 5 prime direction? You may be wondering because I asked myself this for years and it was really hard to wrap my head around. So let's go through it. Firstly, to understand the directionality, the sugar, this blue one here, is the backbone of DNA and it has carbons, which are listed here. Carbons are listed in a clockwise direction. So 1, 2, 3, 4. The 5 prime carbon is actually outside the ring structure. So don't get confused as to why 0 is here and 5 is here. It's not a mistake. The 5 prime carbon is actually outside the sugar structure. It is also clockwise when we number it on the other side. So it'll still read 1, 2, 3, 4, 5, but you can see it's flipped, and that's the anti-parallel nature. So when we visualize this, we say that the left strand here runs from the 5 prime end to the 3 prime end, and the right strand runs from the 3 prime end to the 5 prime end. What does this mean? I must admit, the first 100 times I heard it, I was still confused. It's just talking about how nucleotides link, okay? So each square here contains a nucleotide molecule, and they consist of a sugar, phosphate, and base. Now, we say that this DNA strand was created in the 5 to 3 prime direction because this was an existing strand, and then this one was added on, and then this one was added on, okay? And the way that it gets added on is shown here. Nucleotides attach through phosphodiester bonds, and what that means is that the phosphate group here, which is attached to the 5 prime carbon of a new nucleotide, bonds with the OH group on the 3 prime carbon of the existing nucleotide. One more time. This is the existing nucleotide that we've got. Pretend we haven't built the rest of this strand here. This is the only one we've got, okay? We create a new strand by taking the phosphate group, which is on the 5 prime carbon of the new nucleotide, and we make it bond, or it bonds on its own, with the OH group on the 3 prime carbon of the existing nucleotide. So it gets built that way. And then this one does here, and then this one does here. So now that we have a good understanding of that, hopefully, let's move on to step 3. How does DNA polymerase copy each strand on the lagging and the leading strand? And what does that even mean? Okay, let's go from here. The green molecule I've drawn here is DNA polymerase. And I must clarify, when I've drawn this, I've shown it when it's already synthesized this new strand. This yellow and orange represents the new strand. So this molecule would have started here and then gone in this direction to synthesize that strand. Same with here. It's not starting here, it started here, and then it went and created the new strand in that direction as indicated by the red line, red arrow. And the way it does this, the way it goes, as I said, I should say instead of goes, it adds new bases to the 3 prime end of the new strand. It's important to appreciate when we're saying it's building a new strand in a 5 prime to 3 prime direction, it's not building the new strand in the 5 to 3 prime direction of the old strand. So it's not going from here to here. You have to appreciate that the new strand has its own directionality because the new DNA molecules that we're creating will be anti-parallel to each other as well, okay? So this is 5 prime to 3 prime of the original strand, but then if we consider the new one and the way that DNA polymerase is going, it's going from 5 to 3 on the new strand, which will be the opposite direction of the original one. That was important to clarify for me personally because I always got confused. So if that's a weak spot for you as well, take some time to understand that because it'll put you miles ahead. And you'll avoid many mistakes when you're looking at an exam question and you're having to transcribe the nucleotides that are added during DNA synthesis. It's such a common way to get tripped up. Okay, um, it happened to a friend of it happened to me. I'll be honest. Do your best to clarify that. Now, this is done quite nicely on this top strand here, where the DNA polymerase moves from here in this direction and creates a continuous new strand, right? However, on the bottom strand, there's a bit of a problem. We call the top strand the unproblematic strand. We call that the leading strand. But the bottom one we call the lagging strand, and this is for a good reason. This is the first sequence that we've created on the lagging strand. So DNA polymerase has gone from here to here. The thing is, though, DNA helicase is going to keep unwinding, right? And DNA polymerase is stuck here and it can't go from 3. It can't keep going here. It has to come back from the 5' to 3' end because that's the only direction in which it can function. So what happens is it jumps here, makes a new primer, and then adds new nucleotide bases, adds new nucleotides. And then once it's here, uh-oh, we have to go again back to the 5' end and build towards the 3' direction. This is why we call it the lagging strand, because then we end up with fragments of the new strand, not a continuous one like on the leading strand. We call these fragments Okazaki fragments. And what we're left with essentially is a lot of RNA primase here, right? Now, DNA cannot contain these RNA primases, okay? So the primers have to get replaced with DNA bases since these primers are made out of RNA. And this is where we lead into the 4th and 5th steps of DNA replication. So DNA polymerase will come in and replace the RNA primers with nucleotides. And at the end of replicating, you'll have 2 identical double helix molecules, but these have to be glued together because of the gaps from the jumping ahead of DNA polymerase, right? So DNA ligase will come in, remember the gluing enzyme, and it'll glue the strands together. And then we've got 2 identical double helix DNA molecules, which are these colored ones here, from the original 1 double helix molecule, which you can trace back and follow as the black end. When they mesh back together, we call them semi-conservative because they're no longer the original one, right? There's a copy of one original, black, and one new strand, orange. And that's it.